Perfect. Thank you very much. We'll be giving actually a lecture on, molecular biology primarily, and this is quite just very technical. So try to bear with me. You can ask questions as well if you like at the end. Basically, this is a DNA strand, as you can see on the left, that you have DNA, you have RNA that carries the information, you have the proteins that are made out of the DNA and the RNA information, and you have sensory proteins, and you have sensory proteins and ion channels proteins on the surface of the cells, and then you have action proteins as well. And basically what happens is that communication happens through the proteins inside the cell and between the cells. And how it's all connected, it starts from very very tiny subatomic particles that are called strings. And from the strings, the resonance of the strings creates different elements, and these different resonances basically create everything around us, including us and the universe. And basically a particle is 10 to the minus 38 meters, an atom is 10 to the minus 15, protein is 10 to the minus 9, so they're extremely minuscule. So it goes from the particle to the atom to the DNA to the protein to the cell to the organ. And as you're increasing complexity, you're increasing the gestalt, what we call the gestalt, which is more than the sum of the parts. So these skin cells, each one forms a gestalt. There is a skin gestalt, and then the human face becomes a gestalt. And we can reverse time. There has been Ben Timbret from British Columbia and David Chang from the University of Maryland have developed a mathematical formula for time travel. But it's a mathematical formula still. The problem is that as you're increasing in complexity, then the gestalt is more than the sum of the parts. And because it's more of the sum of the parts, you cannot, as a full person, you cannot go back in time, because going back and returning will be, sort of a bunch of scattered puzzle pieces that cannot be reformed in the same whole that is more than the sum of the parts as it was previously. Now, time reversal is only possible on the smallest scale, and physicists have just reversed time on the smallest scale by using the quantum computer. The conclusion is that if we want, in anti-aging, if you want to reverse time, go back in time, you cannot do it with full people, you cannot do it with full organs, you cannot do it with layers of skin, you can only do it with molecular mechanisms. And the Nobel Prize in 2018 was given to these investigators, which basically looked at the CTLA-4 and the PD-1. They're T-cell breaks, and they postulated that if you can take these breaks away, then the immune system will attack cancer, and they basically, in that sense, they developed a cure for cancer, and that's 2018 Nobel Prize. You see this in all molecular elements, that GDP becomes GTP, ATP becomes ADP, and back ATP. They go back and forth in time routinely as part of their functioning. The genome rearrangements are also showing the sorting situation by reversing themselves and then rearranging themselves. So again, they go back and forth in time routinely. These molecular mechanisms that we cannot see with whole objects, with whole people. You can do reversal by resonance. This is an example, basically scientifically, a time periodic gate voltage example of how that circle turns into a full circle through the resonance. What is resonance? Resonance is energy that you transfer, and the cells work with resonance. They transfer energy one to the other, basically. So proteins, as you can see, on the second protein, there is a difference because that protein, through resonance, has been modified. And resonance is like a hybrid. So you have a donor emission, you have an acceptor emission, and then you have sort of the hybrid, which is what we see the FRET. It's a filter basically that creates that. And this is again very new research. Now, the resonance energy, it's called the Jablonski diagram, and it's talking about how the donor and the acceptor, and as you can see the microscope on the right-hand side, you can see it's a very, very, very complex microscope that you can sort of observe the resonance and how it happens. Now, what is the origin of skin disorders? And the origin of skin disorders is in the DNA. Basically, the mutations in the DNA create a problem. And the chromatin is rearranging the nucleosomes, and the smaller the mechanism, the easier and faster the repair is. And again, here you see an enzyme that has been instructed to basically repair the double helix and repair a broken strand. And I can show you in a movie how this happens. As you can see, this is a DNA polymerase. It's an enzyme or a protein that copies the DNA on the body. And the DNA strands are matched up A with T and G with C. However, if A is matched with G, then this is a mismatch that can cause faulty proteins and nonsense signals, and that can actually lead to cancer. The immune system inspects that, basically sees that, and brings other immune proteins that are combined with other proteins to remove that damaged part of the DNA. And when that is removed, then another enzyme that, again, the DNA polymerase enzyme comes and joins these two undamaged DNA parts to make back the actual DNA that is not damaged. And our body does this repair routinely. This is how you code DNA. It starts with ATG, and TGA is the ending of the sequence. However, if you have one letter, you can see the C here, is replaced by a T, then everything stops and it turns into a nonsense signal. And a signaling correction through resonance, you can basically correct that. You can basically bring these proteins to correct this DNA part. And here comes the problem. The same signal, for example, a nerve growth signal or a signaling pathway can have different functions. So the same signal can also enhance or suppress cancer. So the signals have to be delivered within a specific, discrete time to fit the biological orchestration of the signals. And again, what you see is a broken sequence, and that when you basically fill in the gaps of that signal, you can have a meaningful signal starting again. But in our research, what we do, we basically look at how signaling technology can repair faded or degenerated cellular signals, and also it acts as a mega antioxidant because it cleanses the DNA from free radicals through electrons that avoid the DNA breakage, and also the signals reinstate the DNA signals. There's 5 mechanisms of signaling. You basically create the signals. They have to be transmitted in ultra low proprietary energies that you cannot feel, in which case the electrons controlled amplified via ion channels. The signals are emitted in variable discrete times, and there are multiple resonance signals emitted simultaneously to have a mega antioxidant effect. Now here is how you put the signals together. As you can see, it's not an exact science because we're still looking at clusters of signals, and this is how you kind of form different signals. Then you work waveforms, and basically that's composed by different waveforms of different shapes, up to 10,000 of them, and you do it digitally, and then you translate it into unlimited resolution with other components. And the signals have to be delivered in ultra low energies. There is a book that came in 2008 that actually showed the mathematical formula that proved that at certain very low energies, the ion channels of the cells are opening up and they're receiving signals. And as you can see, it's extremely tiny, the microvolts. And the signals, our signals have to be delivered within discrete times because our cells have a circadian clock. This is again, that's in the suprachiasmatic nucleus. And this is the Nobel Prize 2017, where they looked at the signals and how these signals have biological clocks. This is an example from psoriasis, and this is a time-controlled illness. You can see a stadium psoriasis here, another clinical stadium psoriasis. And basically what it is that the question is, can you unboil an egg? And the answer is yes, because the natural proteins that affect the other proteins that leave this part like a fried egg, basically, a tangled mess. What you can do, you can basically renatural protein, we can refold proteins, and this way that's another kind of avenue to kind of cure cancer. Here is how skin damage can happen through proteins. Autophagy is a mechanism that cleanses the system, and skin disorders and aging are the result of aggregated, unfolded proteins. Basically, an unfolded protein gives nonsense signals, and radio frequency, for example, radiation leads to protein denaturation. That's a denatured product. That means it's a damaged protein. But there is protein refolding. Now there is, it started in 2004, and now it's becoming more and more kind of sophisticated. There's different structures of proteins. The primary proteins is better, easier to refold than the tertiary structure of proteins, and this is the structure of different proteins. And you can actually measure it with a second virial coefficient. A negative second virial coefficient indicates of negative protein to protein interactions. A positive one indicates that there is a protein integration has been decreased. And as you can see, again, the same thing, radio frequency affects proteins, denaturation, but we can actually now refold the protein with different techniques like resonance and cyclic voltammetry. And this is our international research and our board, clinical board. Thank you very much.